Hello students and welcome back to Microbial Concepts. So this is fourth video on citric test, which is a test from IMViC group, and in this video we will be covering few viva questions on this test and the principle and how the test is performed. Okay, so let's start with this. So C in the IMViC, it stands for citrate test. Okay, so already a video is uploaded on Indole, MRVP, that is methyl red, and Voges Proskauer, and this is the fourth test which covers citrate. Okay, so it is a biochemical process which is used to detect ability of organism to utilize citrate as a source of energy, and it is used to differentiate among the gram-negative bacilli in the family of Enterobacteriaceae. Okay, so that is the process, or that is the main biochemical process that we are following here in the citrate test. The bacteria that can grow on this media, it has the, or one that is able to use the citrate source, it has the ability to produce an enzyme that is citrate permease. Okay, so only those bacteria which have the ability to produce the citrate permease, they are capable of converting the citrate, means they will use the citrate and then convert it into the pyruvate. So then pyruvate, it enters the metabolic cycle for the production of energy. So this is how citrate is used as a carbon source to obtain energy. Okay, so growth on this media, it indicates utilization of citrate. Once the bacteria is able to grow, that means it can utilize the citrate. The ammonium salts from the media, they are broken down into the ammonia, and this changes the pH to alkaline. Okay, and thus the media contains the dye that is bromothymol blue, which is a pH indicator. It turns green to blue above pH 7.6. So 7.6, above 7.6, there is, if the salts are broken by the bacteria that can grow on the media, then the pH shifts to the alkaline, and then the bromothymol blue, it turns green to blue. Okay, that's the main reaction we are going to observe. Now for this test, media that is commonly used is Siemens citrate agar. It has pH 6.9, that means it is nearby neutral. Now the Siemens citrate agar, it is made in deionized water. It has sodium chloride, sodium citrate, dehydrate, ammonium salt that we have already seen, that is dihydrogen phosphate, then dipotassium phosphate, magnesium sulfate, bromothymol blue, the pH indicator, and agar. Okay, now this is the reaction. So you can see bacteria that has the ability to produce citrate permease enzyme. What happens? This enzyme, it brings citrate inside the cell. Now the citric acid, it is converted by the citrase enzyme to oxaloacetate plus acetic acid, oxaloacetate to pyruvic acid, and then the pyruvic acid enters the metabolic pathway to gain the energy. Okay, now about the procedure. So the Siemens citrate agar is used, and the slants are made for the citrate agar test. Okay, or citrate test. So you can see here in the picture, slants of Siemens agar, citrate agar are made, and on that you streak your inoculum. Now streak the slant back and forth with a light inoculum which is picked from the center of a well isolated colony. Now the culture that you are using for any biochemical test should be freshly grown, that is max 18 to 24 hours old. Okay, to get your biochemical results precise and accurate. After streaking, you incubate aerobically at 35 to 37 degrees Celsius for up to 4 to 7 days. Okay, then you observe a color change from green to blue along the slant. Now already at pH 7 or 6.9, the bromothymol blue is at neutral pH, it is green. When the pH shifts toward alkaline due to the bacterial growth and change in the pH, the color changes to blue. Okay, so if you get such blue color, then you interpret it as positive, that yes, your bacterial culture whichever you are testing is citrate positive. That means it can utilize citrate. So some examples here. So these are some known bacterial cultures which show citrate test positive. Okay, so they are Salmonella, Edwardsella, Citrobacter, Klebsiella, Enterobacter, Serratia, and Providencia. And negative citrate test is shown by Escherichia, Shigella, Morganella, and Yersinia. Okay, so try to remember at least two examples. And some organisms, they are capable of growth on citrate and they do not produce a color change. So growth is also considered as a positive citrate utilization test, okay, even in the absence of color change, right? If the bacteria is able to grow, that means it is able to use citrate as a sole source of carbon, so it is also considered as positive even though there is no color change. Okay, so some viva questions on this test. So the name of media that is used for citrate test, that is Siemens citrate agar. Examples of citrate positive organisms, citrate negative organisms, we have already seen that. Color change observed for positive reaction, so that is green to blue. In the blue is in the alkaline pH and at the neutral pH it is green. So which is the dye? Dye is bromothiamyl blue. What is the sole source of carbon used here? That is citrate. Okay, then name the enzyme that is involved in citrate test. Okay, citrate permease. So this is about the citrate test and I hope the concept is clear to you all. These are the probable few questions on this test. So if you get IMViC test in your biochemical for your practical exam, then I hope all my 4 videos help you out and you get your concepts clear. Even Viva questions are listed here to help you out. So I hope everything is good. Now you can easily proceed for your Viva exams, for your practical exams and score good. Okay. So like my videos, do share my videos with your friends and do subscribe to my channel. Thank you.